lot of students get confused about this hemophilia mutations homework, so let's go through it together. You'll be doing yours on your computer, but I printed it out so that we can see it together. This first page is just background about hemophilia and the different mutations and what it causes to happen in the body. That will become important at the very, very end of your assignment when we have the DNA sequences and you've got to figure out whose DNA is the most mutated. So we'll get to that later. Then we start back here. Before we dive into the assignment, let's do a quick review. I printed out a codon chart. You don't have to print one out. I just printed one out so you can see it. All right, if we have a sequence of DNA that is GAT, and I were to ask you to replicate it, you would remember the rules that A always pairs with T and C always pairs with G. So here, G, we know it's always going to pair with C, so that's a C. A always pairs with T, T pairs with A, and then C always pairs with G, A will pair with T, and T will pair with A. You just know it by following what would equal what. That's DNA replication. When we get to mRNA transcription, RNA does not have a T. There's no T base. It has a U instead. So you follow the same rules, you just substitute a U instead. So let's say I gave you a DNA sequence that was GAT, CAT, and I said, please transcribe this. Okay, well, G always pairs with C. A would pair with T, except there's no T, so instead it's going to pair with U. And then T pairs with A. C pairs with G. A would pair with T, but there's no T, so it pairs with the U instead. And T, right here, will pair with A. So compare these. These are the same, except you substitute that T for a U. Okay, let's dive in. We'll just do a little bit of some of this. Okay, your original DNA sequence is where we start. All right, we are going to go ahead and transcribe this into mRNA, and they've done the first sample for you. T would pair with A pairs with U, and C would pair with G. Okay. So A, right here, A always pairs with T, except there's no T because it's RNA, so you put a U instead. C pairs with what? With G, and C pairs with G again, and so on and so on. I'll do one more. T would pair with A, T would pair with A, and G pairs with C. You can go ahead and do the rest. The next step is we've got to figure out the amino acids. This is the original DNA sequence. This is with no mutations. This is a perfectly functioning chunk of DNA. We've got to figure out what this stands for. This is where we use our codon chart. We've already done this first one for you, AUG, and here's how we use this codon chart. The first letter, A. So the first letter is A. Second letter is U. So on the chart, second letter, look for U, okay? So where these two line up is in this box right here. And the third letter is G. So AUU, nope. AUC, nope. Here's AUG. It stands for methionine, and you also know that methionine, or AUG, is a start codon because of your PowerPoint. So this is what's starting it. So you can write start or methionine in your work, whichever one is acceptable. All right. Next, UGG. We are translating this now into amino acids. So UGG, we got to figure out what amino acid this codon, that set of 3, stands for. UGG. So first letter is U, second letter is G. These two match up in this square. Now we know these all start with UG. We've got to find the one that ends with G, which is right here, and it's trip. Well, that's the abbreviation. Troponin. All right. The next one we did AAC. Let's see. All right. A is the first letter. A is the second letter. They meet in this little section here, and find the one that ends with C. It's right here. And you just put ASN. And that's how this is done. You do this for the DNA sequence, and then you go down and do this all again. The difference here is you get to highlight any changes as you go. So you'll highlight any changes. Right away, just looking at this, we know that something happened. Because right here, we've got ACC, and here we've got ATT. So hang on. Did we get rid of the 2 C's? Did we delete them? Or did we change the C's into T's? If you look at this and compare the 2 sets, you can tell there's other differences that help you know. This is shorter. This is just one letter floating out here by itself. So this is a frame shift mutation. We know it's going to be a frame shift. We don't know yet if it's an insertion or deletion. But taking a look at this, if we count the number of letters, this has fewer letters. So we know this was a frame shift deletion. And it looks like 2 letters were deleted in this case. And we can double check to see if we're correct. ACC, TTG, all right. A, if we take out the CC, then we still have TTG up here. TTG. So it goes on. All they did for this mutation, all this that happened to this DNA was these 2 base pairs were removed. So as you make your as you get your highlighter out and as you make your transcription here, you'll write, okay, TAC you already know from before. That's going to be AUG, because you've already done that. And you know it's going to be methionine. However, we know this is probably going to be different. So if this is different, when you look at it, when you actually do the work, highlight if there's any changes in here. And then you need to highlight all that apply for this effect on the protein. So was there no change at all? Sometimes mutations don't actually change. You end up with the same amino acids anyway. Cool. Sometimes only one amino acid changed. Sometimes there's a stop signal right in the middle instead of going all the way through, and it just stops coding some of the DNA. And one amino acid added or deleted, that can be different. You might also add if there were two. In this case, there were two, but you could highlight that because at least one was added or deleted. And all the amino acids changed after the point of mutation. So when you do this, do all the amino acids change from this point, or was it just one change? So highlight all the things that are true. You will do this for your 3 mutations here. And then when you get to the end, what you have to do is read about these case studies. And basically, you have to figure out who has the worst hemophilia. Which mutation got the least amount of the good gene working? Because we want this whole good gene to work. But if only part of it's working, you're getting some benefit from it. But if the whole thing's not working at all, then you have it worse. So you're going to read these people and try and figure out, based on your reading on the very first page and based on what you've discovered on your way through, you've got to figure out who's got what, who has the most severe hemophilia, and which mutated DNA sequence they likely have.